Let's say that in an embryo there are a bunch of cells. Do those cells know what organ they are going to end up as? For example, if a cell is going to finally have its fate as being a part of brain, does that cell know that it is going to be a part of brain in future, and so for the other organs and tissues? And if so, or what you can say, if they do know, if the cells do know that what are they going to become as the developmental progress, as the developmental process will proceed, then how do they know? And when do the cells realize that they are going to be part of a specific, a certain organ? What in other ways we are trying to say here is that a cell, well, that is undifferentiated, let's say an undifferentiated cell in embryo, it is going to be somewhere in the embryonic development or in the fetal development is going to be end up as some particular cell. But for that to occur, the cell has to go through a maturation process that is divided into what you can say two stages: the cell commitment and the cell differentiation. Cell commitment is itself divided into a couple of stages: the cell specification and cell uh, termination. And first of all, a cell is specified to a certain cell lineage. Let's say muscle cells, and that cell that has been specified to be a part of a certain cell lineage, that is then going to be determined to be a certain cell. And after determination has occurred, then the specific pattern of gene expression, they are going to cause the differentiation of that cell that was determined to be a certain cell to be that certain cell, or what we call differentiation into that certain cell. Let's say a cell, first it was specified to be a neuron, then it was determined to be a neuron, then it differentiated into a neuron. In the course of that, all those processes in which a cell realizes or knows what it is to become in the future, the first thing that we need to know is cell differentiation. And this cell differentiation, it means a generation of specialized cell types. And in this, a cell ceases to divide and develop specialized in structural elements and distinct functional properties. So this is what the cell differentiation means. Means no more division, no more proliferation. It is just going to be the development of specialized structural element in the cell that are going to lead to the distinct functional properties of that cell. And it is what you can say the last stage of the series of events that commit at an undifferentiated cell of an embryo to become a particular cell type. For example, a red blood cell, obviously it differs radically in its protein composition and cell structure from a lens cell in our eye or a neuron in our brain. But these differences in cellular biochemistry and function, they are preceded by a process that commits the cell to a certain fate. It means that, in simple word, there is going to be some process that is going to tell this cell to end up as being a red blood cell with a particular, what you can say, structure, with a particular biochemistry, and we are going to, what you can say, discuss that process in brief. And during this course of commitment, or what you can say, when a cell is committed to a certain fate, that hey cell, you there, you are going to become the red blood cell, and you are going to become another cell, you are going to become the neuron, and you are going to become the muscle cell. So this commitment of cells, uh, but you can say that it has a couple of stages. It can be divided into two stages. The first of the two stages is the specification or cell specification. And in this, the fate of a cell or tissue, it is said to be specified when it is capable of differentiating autonomously or by itself when placed in an environment that is neutral with respect to the developmental pathway. Or what we can simply say is that after the cell specification has occurred, the cell knows that it is going to become either a neuron or a muscle cell or a red blood cell or a bone cell or other cell, etc. And when we place that cell that has been specified alone in what you can say a petri dish or test tube, that cell is going to differentiate autonomously or by by some changes in its own self to become the uh, cell that it is specified to be. For example, a cell that has been specified to be a neuron, it is going to get differentiated into neuron when it is placed in a neutral, what you can say, environment, or the medium that is, or the environment that is not influencing the cell to change its specification, because the specification is reversible. At the stage of specification, the cell commitment, it is still labile, or what you can say, capable of being altered. So if you change the environment of cell, then the cell's uh, specification it may change. For example, a muscle cell, when it is placed in a bunch of a cell that is specified to be a muscle cell, when it is placed in a bunch of cells that are supposed to be uh, nerve cells or neurons, this muscle cell is going to differentiate into a neuron rather than into a muscle cell because of the environment. So if a specified cell is transplanted to a population of differently specified cells, the fate of transplant will be altered by the interaction with its neighbors. And the second stage of committing a cell to a certain fate, it is the determination. And it is not label, it is not what you can say, it cannot be changed. Once a cell is determined to be a certain type, it is going to be of that type regardless of the environment. If a cell that was earlier specified to be a nerve cell, it has determined to be a nerve cell. Now that nerve cell, regardless of the environment it is in, it is going to be a nerve cell. And a cell or tissue is said to be determined when it is capable of differentiating autonomously even when placed into another region of embryo or a cluster of differently specified cells in a petri dish. So regardless of the environment, once a cell is determined to be of a certain of be having a certain fate, the cell is going to have that fate. And if a cell or tissue of is able to differentiate according to specified fate, even under circumstances, it is assumed that the commitment is irreversible. So now, after the second stage has come in the commitment of cells, now the commitment has gone irreversible. There is no way back. There is no way that a cell that has determined to be a neuron is going to become a muscle cell in any possible. So we can say that during embryogenesis, an undifferentiated muscle cell, it matures through specific stages that cumulatively it to a specific fate. The first of the stage is self specification, and then the determination, and finally the differentiation. So first, the cells are specified for a certain fate, then they are determined to be reaching that, then they are determined to reach that certain fate, and finally they are differentiated to become that cell that they were determined to be. And during specification, there are three major strategies, but we are going to discuss each of those strategies in individual separate lectures. But let's take a look at what we just talked diagrammatically. So let's say that here we have what we have. Here we have a blastula. And in this blastula, let's say it is a brown cells. These are specified to be muscle cells, and these yellow cells they have specified to be a neuron. And when they are placed in neutral medium, they are going to be ending up as the cells that they were specified to be. So they are going to be first determined to be muscle cell, and then they are going to be differentiated into muscle cell. And in this yellow cell, it is first going to be, once it is specified that it is going to be a neuron, it is going to determine to be a neuron, and then it is going to be differentiated into a neuron. However, if this brown cell that was specified to be a muscle cell, it is placed in a bunch of nerve cells, or this yellow cell that was specified to be nerve cell, this brown cell that was specified to be muscle cell, it is going to change its specification, and it is going to become a nerve cell rather than being a muscle cell. It is going to end up as a nerve cell when it is placed in the environment of nerve cell. However, when determination has taken place, then there is not going to be any reversibility possible. For example, let's say that this cell, this brown cell, this muscle cell, uh, it was specified to be a muscle cell, but then it gets determined to be a muscle cell. then regardless of the environment you put in, let's say that after the determination of cell had occurred, and after determination you put this cell that was determined to be muscle cell into this cell that are determined to be neurons, then there is not going to be any effect. That muscle cell is that cell that has determined to be a muscle cell, it is going to end up as a muscle cell, being a muscle cell, being differentiated into muscle cell. And the neurons and the cells that were determined to be neurons, they are going to differentiate into neurons. So this was all about this, some basic definitions from where we had to start this. And in the upcoming lecture, we're going to take a look at the three types of the cell specification in uh, detail. I hope that you have found this video to be beneficial and of appreciable quality as well. And if it is so, then please do like this video and do not forget to subscribe and turn the notifications on. Thank you so much for watching. See you in the next lecture.